Alright, I'm going to help you out in finding some of these sequences. There'll be three sections like we have seen before, or three tasks, and each individual will work on each task. And I'm going to do one part of each task for each person. So the one thing we need to know is we have to have these IUPAC symbols for what the various letters mean. That has been posted as a announcement, so you can find that on D2L. Now, moving forward here, let's go look and see what we've got. I have my basic sequence here that I actually don't think does anything, but that's not the thing here. You have to essentially follow what I do. I don't think I'll find anything, but because I don't think this is a gene, but maybe it is, because we found a few things the last time I tried it, so maybe we'll find something. So here are my IUPAC, and what you're going to have, in addition to you're going to have your sequence, but you're going to have these set of rules, which I'm going to post. So for task A, what I'm going to do is I'm going to help you find the TATA. Now, the people in task A are going to be looking for the core promoters. In other words, or the core pieces that control transcription and allow it to operate. So there's going to be a TATA box, an INR, which everybody will find an INR. For one thing, it'll tell you where your transcription start site is. You may have more than one INR. You have a GC box, an OCT, and a CAT box. So I'm going to find the TATA box for task A. So the people in task A have to find TATA. So how do you find TATA? Well, a TATA is this sequence. All right, and I'm going to make some space here. And so let's go figure out what a TATA actually looks like. It's a TATA, yes. So that's good. We know those letters. All right, but what is this W? Well, that's an A. We know that one. But we have another, we have another W and then an R. All right, so let's go over here to our IUPAC rules and find out what the W's and the R's are. So we know GATC right there. An R, it's an A or a G. All right, so let's go back over here and let's put R as A or G. A or G. All right, and then what is W? Well, W is a or T. So that means A or T. We can go back over here. A or T. Now, this isn't set in stone. Just because one or two letters are off doesn't mean this isn't a TATA box. You're finding the most likely place for a TATA box. Now, it's supposed to be at -30. That means -30 before your transcription start site. So if we find this somewhere, or we find a series of them, you can have more than one TATA box. And if they're all around -30, then you can look forward for the TSS about 30 base pairs. So this is a way to assure yourself that you found the transcription start site, or at least one of many transcription start sites maybe, by its location to the TATA or TATAs that you find. So let's do this. I am going to start with, we always start with TATA. Then we're going to what? We're going to find an A or a T. That's what we want to look for. Then we get an A. Then we get another A or T. And then we get an A or a G. All right, just to kind of know what we're looking for right here. So, of course, when I go look through our gene, let me go up here. All right, what do I want to look for? Well, we're going to look for TA. Now, I want to just look for TA first. All right, and I just want to see if they're clustered anywhere, and they're really not. All right, maybe down here. but no TAs really up here where I'm expecting my promoter. So, not real good here. So then, oops, what is my next thing to look for? A TA. I'm going to go ahead, or let's just add the T first. Let me go back over here. Let's just see how many of them have a T here. Well, here's one, and it's up at the beginning. All right, so what's the third letter that, maybe this is it. This is our TATA box. All right, TAT, and then TAT, an A or a T, right? Nope, that needs to be an A. So it's TATA, A or T, and then A. So what do we have? TAT, but no, it should be an A there. It should be, according to this, TATA. So it should be, if it were strict, TATA. And we don't find that. We do find TAT here, and then another T. But you know, if you look at this, oops, I keep going back to there. If you look at this, the only place you see a G is at the end. This is a series of T's and A's. That's really what a TATA box is. It's a series of several T's and A's in a line. So really what you should look at are A's and T's. So let's look at this in the big picture. What should we be looking for in this sequence? Well, we should be looking for things like TA. We go back and we look in here, and do we see anything that looks, no, you get these C's and G's close by. Here we're going pretty good, but then we get the G's and C's. Here we're going good, we've got a C. No, that doesn't work, that doesn't work. None of these actually make for a good TATA box because you end up with some C's and G's after that. Let's try a different sequence. Instead of TA, let's try AT, and then let's see if we do any better. Here's an AT, but nope, we stick a G there. AT, there's a G. Oh, ATT, nope, G's. AT, no, AT. Let's try, this looks a little better, but let's try ATA. Nope, G. And so now let's try ATT. Nope, nope, no, no, no. I don't think this is the best one right here that we've seen. It's 4 A's or T's together. We could try something like this. We could try AA. Nope, nope, no, no. Not in the regions I'm wanting it. You always get a G or a C. There's 3 A's together. You really don't get it. So let's try 2 T's together and see if we get anything. No, 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 no. It just doesn't happen in this sequence, especially not in the regions that we're looking. This would be fine, but there are 2 C's in between. And so my conclusion here is this is a TATA-less promoter. All right. So you realize what I did, and when you really look at the idea, and this is why you can't really program this in sometimes. Sorry. Realize it's, well, you can. You could write this in a computer program to search for this, but I don't know how well it would do. The whole idea here is you have a series of T's and A's. Looks like 4, 5, 6, 7, 8, 7 or 8 A's and T's in a row, and then potentially a G at the end. All right. Otherwise, it's all A's and T's, and we just don't find that in this one. So in my sequence here, I would say this is TATA-less. All right. So this should give you an idea on how to actually find a TATA box in a eukaryote. All right. It's more really of not finding the exact sequence. It's finding a series of A's and T's that go on for, I don't know, at least 5 or 6. All right. And then maybe you can have a G in there. That looks like what this is, a G or a C. Actually, it doesn't look like C's occur ever. Maybe a G or a C at the end, if any. But what you're looking for is a series of A's and T's. They don't have to be TATA. You know, the most common TAA, TA, You know, that's I believe the most actually the most common found functional TATA box, and you don't find that in here. And I don't find this one. I do find a TAA. So here's a TAA, CA, just a little bit too much. No, there are no TATA boxes here. All right, I just don't see any TATA boxes. Now we could try it backwards. That's always a good idea. Instead of TATA. So we did find an ATT. We looked at this, I believe, and nothing. You always get that C or a G, and they're up too close. There are no TATA boxes in this one. All right, so this is a TATA-less promoter. All right, so that's what you need to do. All right, and so go look at yours and do TATA. So person who has task A, you should do TATA INR, which should be right on top of the transcription start site. So if this one's +1, you should be, if you have a TATA, it should be about 30 bases upstream of it. That's kind of coordinating. Now GC boxes are a long way away, but they should be kind of on the outside of your, kind of when you're getting the reduction in your CPG island. In other words, maybe about where your CPG island begins. Maybe you start looking for it, but it could be anywhere. Your CPG island may be 600 base pair upstream, but 200 is getting pretty far out there. You're out of the core promoter probably, and you're moving into the proximal promoter right there where the GC box is. The oct, this is another one, should be at -50 to -70. Doesn't have to be, and it doesn't have to be exactly this. All right. Ooh, this looks like a sequence we were looking at. No, I don't think we saw 3 T's ever. I was going to say maybe it has an oct, that one I was looking at. And then a cat box, all right, at -100, so 100 base pairs upstream. All right, so that is what we want for task A. This is probably the more important one, is finding the primary promoter sequence. One of these is needed. Well, no, I shouldn't say that. There are probably about a dozen known. But these, I would say you're going to get 95% of all genes are going to have one of these 4. All right, so for RNA polymerase II. If you're using RNA polymerase II in a eukaryote, you're probably going to get one of these 4. Insects can be a little different. Plants are also reasonably good with this, so it should work. This part should work reasonably well in an algae. Of course, no one has ever studied EHUX that well, and EHUX is a bit of a strange organism, so you never know what you'll find. All right, so task B, let's do this for, let's see, which of these are good? Now there are 1, 2, 3, all of these are DPE, so there are 3 sequences associated with DPE, and they are in the upstream, the untranslated region, which means they're after the transcription start site. So these are actually would be found in the RNA. Now remember, you need to keep track if you're an RNA or DNA. If you think you're an RNA, maybe you should translate these back. But look to see if you can figure out if you can find these. We'll do another exercise for it. Just look for them. You'll need to translate each of these. So I'm going to do what? M3. It doesn't give you a location for M3. So now this one just means BRE up, means it's upstream before the TATA. So if you find a TATA, you need to be looking for this one. So this person might need to coordinate a little bit with this person to tell them, hey, I didn't find a TATA, which means you probably aren't going to find these. All right. Now, if you do find a TATA, then you should definitely be looking for these. This one would be before it, and this one would be after it. So this up means upstream or before the TATA. This one is downstream after the TATA. And usually, you don't usually find both, I don't believe. It's in the PowerPoint I gave you. But you could find, actually I don't remember, maybe you do find both. Look it up. But the one is up and one is down. Look for both of them and report if you find them. Review the PowerPoint, and it'll tell you more details about BRE up and BRE down. They're just around the TATA. So hopefully you find a TATA. You can find these too if they're there. And then this one is also in the UTR. Actually, we don't know where these are. These are in the UTR though. So there are 3 of them relative to the transcription start site. That's why they're plus here. They're what? They're past the transcription start site. They're actually could be found in the RNA. So these are DPEs. Let's see, which one of these do I want to find? Let's just look for the CGANC. That looks pretty easy. Let's see if I have one of those. So CGANC. Oops. So that was CGA. CGA. Now it's an N. That's not going to be found. Wait, I'm in the wrong one. Okay, so make sure I'm at the beginning. All right, so CG. Of course, we find CGs. CG can be methylated, realized. I keep going back to that. CGANC. See if we find any CGA. We do. And CGA, so anything and a C. Anything in a C? No. Anything in a C? Anything in a C? Anything in a C? No, it's kind of close there, but anything. Ah, look at that. Right there. That might be an upstream signal. See anything in a C? Anything in a C there? No. Anything in a C? No. Anything? Oh, here's one. It's a potential one. All right. So report if you find any of these. All right. And I found 2 right here in mine. Here's a CC, so that would work. So, CGA, anything, and then a C. And it should be 18 to 22. So if I find this, I would go back, and since this is supposed to be in the untranslated region, I would go back and look maybe 20 base pair before and see if I don't see signals for an INR or something like that, because the INR surrounds the transcription start site. And so that would be a way for you to confirm your INR, would be to find this about 20 bases after your INR. And then the INR, of course, would be what? 30 after the TATA. So if you found where you think you have a TATA, and then you have an INR about 30 later, and then about 20 later you find this one, you are feeling very confident that you have figured out what's going on in that promoter region. So these 2 people, someone will have to find these. The TATA and the INR would be a good thing to find. Then you would go and see if you can't find these somewhere, after the INR, with the TATA being before the INR. Now the last person here needs to find the DREs. Oh, I never did put the DREs down here. Let me write those sequences down real quick. And that's TAT, so it looks kind of like a TATA. CG, ATA, and then the other version of it, which is only found in certain cell types, is similar. TAT, CG, AT, and then maybe a C. So I'll put a little C. All right. So here is the DREs. They're found in a specific pathway that's very important. P51 is your suicide gene. It's the gene that starts off the cascade for the cell to kill itself. So this one is highly regulated, and everything in this pathway, it tells the cell to lyse, to essentially burst. All right, and so this one is usually turned off. All right, and you have to regulate it. This pathway that kills the cell, you don't just turn this one on just anyhow, and it has some specific sequences that control that. So the DRE is one. These are safety, like putting the safety on a gun or something. If you have one of these in your gene is dangerous, and that tells you something about it. So let's see if we can find one of these. Let's see if we can find the first one here in our gene. So it's TATC. So I'm pretty sure we found some TATCs or something similar to that before, didn't we? So I do TATC. No, there's not. There's no TATC here. So there is a TAT. Let's see if we can find a T. I'm just going to change it up a little. TATC. Nope, don't find anything. Let's try an ATC. We do find those. So let's see if we can. I'm just kind of fooling around here to see if we can find something similar to that. So I found an ATC. Let's see if we can find a GATA after that. So G, yeah, A, ooh, T, no, it goes away. All right, so didn't find a suicide gene. This is not. If there's a gene here, it's probably not not something that is dangerous. So you found some more out about your gene if you find these particular sequences. All right, so that's kind of how you do this. Make sure you look at this. This information is also, I think I gave it as a handout early in the semester, and then I posted it recently as an announcement. So here are your, I will put this in an announcement, this information right here for the next 3 tasks for you to operate on. We're getting close to the end. There's just a little bit more to go. I'm going to have you watch a movie, and we're pretty much done with the semester. So we're getting close, 2 or 3 weeks, less than that, 2 weeks maybe, and you'll be done with this. All right, so let me grab this right now, and I'm going to go post this on the announcements. So good luck. Email me if you're having any kind of problems whatsoever. Email me, let me know what's going on. A few of you have. I've tried to help you out. You've figured out some things. And then after this, we'll get one more task in, and I'm going to have you look at the other strand of the DNA. And once we're done with that, you can write your full report, pool your information, see if things are working out the way you think they should. See if you actually have a real gene there, and maybe tell me a little bit about what it might do.